All right, let's dive right into a huge mystery that lives inside all of our heads. Today, we're exploring some absolutely groundbreaking science that's revealing the secret cells responsible for carving our most powerful experiences into our minds, forever. And it all starts with a question I'm sure you've asked yourself.
Seriously, think about it. You know the ones I'm talking about. Your first kiss, maybe the shock of a scary accident, or that one moment of pure, unforgettable joy. Some memories just feel like they are burned into your brain, right? Meanwhile, you probably can't even remember what you had for breakfast yesterday. So what gives? What makes an emotional memory stick so hard?
Well, to answer that, we've got to look deep inside the brain, all the way down to the cellular level. For decades, the story of memory was really a story about just one specific type of brain cell. But it turns out that story was missing a huge, crucial character.
For ever, neurons have been the undisputed stars of the show. They were seen as the architects of memory, the cells that would fire together and write the first draft of our experiences. But new research has revealed this surprise helper, another cell that takes that first draft and decides whether to engrave it as the final, permanent copy.
So the old school thinking went something like this. A memory exists as a physical trace, a network of connected neurons called an engram. This was the blueprint. But scientists were always a little stumped about how this blueprint gets stabilized. You know, how it goes from a temporary sketch to a permanent structure you can recall years later. And this is where the story takes a huge turn. While they were studying these engrams, researchers found a surprise witness at the scene, a cell that was pretty much ignored for years, but, as it turns out, is the real key player.
Meet the astrocyte. Its name literally means star cell, which is pretty cool. But for years, scientists saw them as just the support staff for the real stars, the neurons. You know, just bringing them nutrients, cleaning up their mess. Well, it turns out their role is way more important to the story of how we remember.
And look, this isn't just some minor tweak to the textbook. As neuroscientist Maite Solis-Subiare says, this discovery is revolutionary. It's shifting the entire focus of memory research from being all about neurons to maybe being all about these incredible astrocytes.
Okay, so what's the proof? How did they figure this out? The evidence comes from this really clever and elegant experiment where they created a strong, scary memory in mice and then just watched to see which brain cells lit up. It's a classic two-day setup. On day one, a mouse goes into a specific cage and gets a mild, but pretty unpleasant foot shock. So it learns to be afraid of that cage. Then on day two, it's put right back in the same cage and its brain recalls that scary memory. Simple, right? And this is where it gets wild. Here's what they found. During that first scary event on day one, the astrocytes were basically silent. But on day two, when the mouse remembered being scared, the astrocytes lit up like a Christmas tree. This was the big aha moment. So let's break that down, because this is the absolute key. The neurons, they were active on both days, when the memory was being made and when it was being recalled. No surprise there, but the astrocytes were only highly active during the recall. That specific timing meant they weren't just forming the memory, they were doing something else, strengthening it, solidifying it.
So that's the what. But what about the how? How in the world do these astrocytes pull this off? How do they know which memories are important enough to strengthen? Well, the answer lies in this really clever two-step process that actually happens over several days.
The secret ingredient is a chemical called noradrenaline. You can think of it as the brain's red alert signal. It's the fight-or-flight chemical that floods your brain during really intense moments, good or bad, basically screaming, "Pay attention, this is important."
And this chemical leads to a two-step engraving process. The first emotional event, that initial shock, doesn't really activate the astrocytes. Instead, that flood of noradrenaline primes them. It tells them to start building more docking stations, more receptors for noradrenaline. They get more sensitive. Then, when the second event happens, the recall, the next wave of noradrenaline has way more places to land. And that's what triggers the astrocytes to jump into action and lock that memory down.
And get this is not an instant thing. It's not like flipping a switch. This whole priming process takes time. After that first experience, it takes 1 to 3 days for those new receptors to reach their peak. Then, after about 2 weeks, things go back to normal. This makes astrocytes the perfect multi-day trace, a system designed to link emotional events that are related but happen days apart.
So why does all this matter, beyond just being a really cool piece of science? Well,
Understanding exactly how our brains decide to solidify memories opens up some incredible new doors for treating conditions where memory goes wrong.
I mean, think about it. In PTSD, traumatic memories are basically overstabilized. They're stuck. What if we could target this astrocyte mechanism to gently weaken that connection? And for something like Alzheimer's, where important memories fade away, maybe we could find a way to boost this process to help lock them in. This gives us a completely new target for future therapies.
So when you boil it all down, here's the big takeaway. Astrocytes are not just the brain's support staff. They are the brain's curators. They actively listen for those chemical signals from our emotional experiences, and they decide which ones are important enough to be engraved into our long-term memory.
And that leaves us with a fascinating, maybe even a little unsettling, final thought. We are starting to understand the exact molecular machinery that cements our most powerful memories. As this science moves forward, you have to ask, if we could actually control this process, what memories would we choose to strengthen? What would we weaken? And what would we choose to rewrite entirely?